Hello again, everyone. This is Dr. West, and this is Lecture 2, and we are going to be talking about the visual system today. So if you remember from the last lecture, we are going to be going through sort of this overall sensory pattern. We're going to start at the bottom. We're going to talk about the receptors, and again, for the visual system, they're going to be located in the eye. We'll then talk eventually about how those receptors will take the energy from the world, in this case, the light energy, convert it into action potentials, which is the only thing that the brain can understand, send those action potentials via neurons up through the nervous system, through the thalamus first, and then that thalamus is going to send it to the primary sensory cortex. And again, if you remember back to the first unit, that primary sensory cortex for vision is the occipital lobe. Pretty much the entire occipital lobe is exclusively dedicated to understanding and processing visual information. And then we will also be talking a little bit in this lecture about some secondary areas that process vision, as well as some association cortex. Now, also from lecture 1, you ended with a video from Crash Course that was going to summarize the basic concepts of sensation and perception. And he also threw in a whole bunch of other terms that you don't necessarily need, but that are pretty interesting. And so what I wanted to do also quickly is take a step back and review. Again, some of you did really, really well with the first exam, and I think you probably used your study tools correctly. But I thought it might be helpful for everybody to see sort of how I would use the study guide if I was a student. So I'm going to take a quick step back. I'm going to go over to the study guide for just a minute and sort of talk to you through how I would use the study guide. So I said I'm going to jump out of here just a second, head over to the study guide, and then I will come back here. So I'll see you back here in just a minute. Okay. So I am on module page 2.1. If you haven't gone there, I highly suggest that you go there. You can download this document. You can do it like I'm doing and just view it online, but you can actually download it and print it if you want, and then you can write your own notes on it. But these are the things that we're going to expect you to know for the second exam. And seriously, it's super detailed. I mean, it's going to have every bit of information that I'm going to want you to know. All right. So again, let's go through, and I'm just going to do the vision part, but this is going to be kind of the level that I want you to know. So let's think back to what you already saw in lecture 1 for unit 2, and then where we're sort of headed. So again, remember, we talked about sensation and perception. And we said that sensation, you really don't necessarily consciously experience, but it's the first step and it's the important part. And all it really is just your sensory organs taking that incoming stimuli from the environment. So the light energy, the moving air into your ears, the chemicals that go into your nose or to your mouth, there's something that's touching your skin, and converting it into an action potential. So they are specialized, your sensory neurons are specialized neurons, right? That the dendrites are modified in such a way that what they interact with is some kind of energy out in the world. So they detect and convert environmental energy, and in case of what we're going to do first, it's going to be light, and it's going to convert it into that action potential or neural activity so that it can be sent to the brain, because the brain itself can't interpret light. I can shine light onto my brain all that I want. It's not going to do a darn thing, right? I have to shine it onto the specialized receptors so that they can then produce an action potential, and then those action potentials can make the brain do a bunch of things, right? You're going to find that we're going to talk about for each kind of sensory system that there's only a little specific kind of energy or a specific range or band of energy that it can respond to. So for vision, there's only a specific kind of energy that it can respond to. There's a specific kind of light that it can respond to. So for example, our eyes don't respond to UV light, right, or infrared light. Our eyes only respond to what we call visible light. We'll talk about that. For hearing, for the auditory system, we only respond to air pressure, which is a form of mechanical energy, not injury. But the somatosensory system, or our sense of touch, is also a form of mechanical injury. So it's when something physically touches our skin or moves the hair that's on our skin or actually cuts the skin. For taste and olfaction, which is a fancy way of saying smelling, it's chemicals that actually enter your nose or enter into your mouth and interact with receptors that are in there. So again, it's a very narrow band of energy. And we also said that each sensory system has very specific receptive fields. So they're only paying attention to certain specific parts of the world. Right? And so if that energy is in that specific part of the world, it will interact with those receptors, and then you'll be able to detect what's going on there. But really what that means is that you're only getting it a small piece of the information that's out there in the world. In other words, there's a lot more happening in the world than you're actually detecting, and you're actually converting into action potential and sending to your brain. But that's okay, that hopefully what you're able to do is, first of all, pay attention to the things that you want to pay attention to, but also take in information that you need to take in information about, right? And that you have more sensitivity on places that you need to be more sensitive, right? So as humans, we need to be pretty sensitive about our sense of touch on certain parts of our body, like our face and our hands, because we use those more, or it's more vital that we be more sensitive on our face, that we protect it a lot more. Right? We don't need to have as sensitive a sense of smell as, say, some other organisms do, like dogs or rats. We talked about that. Right? So we don't have as many receptors in certain places as we do in other places. Right? We talked a lot about this pathway, so I can guarantee you this pathway is going to be part of the exam. We talked about how the sensory information comes into the receptors, and that part is the sensation part. And it's the job of these receptors to convert it into action potential. And then it's sent to the brain, and that part is called encoding. Encoding is literally converting it into action potential, and it's sending it to the brain, and that's called neural relay. Right? Basically just means like a relay race, passing it from one to the other. Neural means neurons, so it goes from these specialized receptors, which are specialized sensory neurons, to other neurons, eventually to neurons in the thalamus, and eventually to neurons in the primary somatosensory cortex. And then that's where the part that you actually consciously experience happens. That's where all the cool, sophisticated stuff that our brain can do actually happens, and that's perception. And that's where your brain does all the interpretation stuff. What is this? All right, which sensory system is it? Am I hearing something? Am I seeing something? Am I smelling something? And then all the cool stuff. What is it that I'm seeing? What is it that I'm smelling? What is it that I'm hearing? All right. Or even more sophisticated, does this food that I'm smelling smell good? Does it smell bad? Does it smell like it's spoiled? Right? Does it smell like it always smells? Does it smell different? Whatever. You have a lot of different things that you can perceive. But most of what you feel like you're going to experience in the world is actually the perception part. You really don't notice that sensation has happened unless you're not getting the perception part. Right? So that's really what we're talking about there. So that's what hopefully you got out of that first lesson. So even though you may have heard some other stuff, all right, in that video from the crash course lecture, all right, this is really what I want you to know. So this is what I would focus on and make sure if you're making something like flashcards or something that you know. All right, today as we move into vision specifically, and again, you got a little bit of preview of it from the crash course video, all right, when we talk about vision, right, we're going to actually talk about the details of how it works. So we're going to talk a little bit about the light energy itself and how it actually goes through a part of the eye called the pupil into the actual eye itself. We're going to talk about the retina, which is the back part of the eye. We're going to talk about the specialized sensory cells that are called photoreceptors. We'll talk about the range of energy. We'll talk about how those receptors, and this is that relay system I was talking about, are going to go to the thalamus, into the primary visual cortex, secondary visual cortex, and association cortex. We're going to talk about a bunch of different kinds of cells on the retina, but I'm going to have you know 3 different kinds. 2 of them are photoreceptors. One of them is also called the retinal ganglion cell. We'll talk about that. We'll talk about what the optic chiasm is. We'll talk about some stuff about the visual field. We'll talk about the occipital cortex, which is also the same thing as the occipital lobe. The entire occipital cortex takes up the entire occipital lobe. There's a bunch of different regions. We'll talk about the primary visual cortex. We'll talk about how information is then sent to secondary regions in both a dorsal way and a ventral way, which gives us different kinds of processing. And we'll talk about all the other cool stuff you can do, like see in color, which is something very unique to some organisms. Humans are able to see a lot of different kinds of colors. We will talk also a little bit about some different theories about why we think we're able to see in color. We'll talk about some specific structures of the eye. We'll talk about the photoreceptors in a lot more detail. And again, I'm telling you here, there's a bunch of different cells in the back of the eye. I'm going to want you to know 3. The rods and the cones, those are the photoreceptors, and then the ganglion cells. Sometimes they're called retinal ganglion cells. All right. So again, use this study guide. So even if you hear more things in the lecture, use this study guide. And then here are some of the kinds of questions. I might ask you to match something like the parts of the eye and what they do, or the kinds of the cells and what they do. I might ask you to label, for example, like label the eye, the blind spot, the retina, something like that. The occipital lobe, I might have you label that again, right? I might have you talking about these pathways, the order of transmission in the retina. So there's several things that I could have you actually talk about. I could have you talk about the 2 different color vision theories, that sort of thing. So again, please take advantage of this study guide. I know I went into a lot more detail about this, but again, I don't think I don't think a few of you really used this study guide on the last exam. I think you tried to study every single thing. And yeah, there's a lot. I get that. Right? But that's why we made this study guide, so we could show you how to sort of narrow it down and what things to focus on so it wouldn't feel quite as overwhelming. So again, hope that helps. I'm going to go jump back over to the notes now and we'll talk about vision. All right, so let's jump into it and let's talk about the visual system. First thing is your brain and your visual system are capable of doing some pretty cool stuff. All right, the first thing is, again, you're not noticing the sensation part, but your brain is able to do some cool stuff, right? Including actually play tricks on yourself. You can do some pretty cool illusions. So one of the first things over here is, do you see a cup or do you see 2 faces staring at each other? All right, or can you see both? Most people can actually see both, right? This one's pretty neat. You have to sort of stare at the middle of it, right, and kind of relax your eyes, but some people say, well, it will look like it's spinning like a fan, right? It's not actually moving. It is actually a static still image, right? But what's neat is that your brain can add movement to it. All right, so that's the other cool thing that your brain can actually do, is add movement. It can add characteristics to something that's there, which in some cases can be neat and in some cases can be very confusing. I love this one. I always ask people, what do you think is here? Tell me what shapes that you see. All right, and some people tell me they see a star, which they're looking at this here, right? And I'm like, okay. Some people say, no, it's not, it's 2 triangles. I'm like, okay, I can see that too. Some people tell me they see a square, and I'm like, yeah, okay, I see a square. Some people tell me they see circles, or they even call them Pac-Man. I'm like, all right, I can see that. And I say, but here's what's really cool about what's here. The only actual shape that I would actually argue is here, you could probably argue that maybe the black square is there. But the rest of them are not actually there, unless you consider Pac-Man to be a shape, but that's, in my mind, not actually a shape. But the circles aren't actually there. These are not actually circles. This is not actually a star. These are not actually triangles. Your brain filled in the pieces, right? Because it assumed depth. It assumed that, for example, this black triangle was sitting on top of the red one, and that's why you just didn't see these lines. And so your brain assumed, oh, well, the line is just underneath it, so that makes it a triangle. And all of that is sitting on top of the red circles. So again, because you're used to seeing things in 3 dimensions, your brain had no problem just filling that in or just kind of assuming it was there. And so you just assumed that those were circles and triangles, or you assumed that it was a star or whatever it was. So you're able to add in details that may not even be there or that may be hidden underneath because you understand the concept of depth and your brain is used to processing that. So again, your brain can add a lot of cool details even if that information isn't being sent from your actual eyeball. So sometimes you can even perceive things that aren't actually being sensed and sent to your brain. And then finally, as another cool illusion here, I love this one. All right, what do you see in this picture? Well, all right, the obvious thing, hopefully to you, is that you see a forest scene with a guy that's riding on a white horse, right? And then having a second horse that's brownish in color following along behind that. That's usually what people see as the obvious thing. But just for fun, later on, I want you to see if you can go through and find faces, right, in this picture. And I'll give you one as a hint. Right, here is one face here. So here's the mouth and the nose and the eyes and like the eyebrow or the forehead. All right, there's one face. The most we have ever found, and I say we, and one of my classes has ever found, is 11. I honestly don't know how many are in this picture, right? But there's at least 11, right? So at some point, see if you can go and find them. And the reason I bring this up is because this is also a skill that most people are really good at. And if you do remember that crash course video, you remember that there is a specific brain region in your brain that its sole purpose is to detect faces, right? And to remember them. And if you remember, that was what he was talking about in the video, is that some individuals are really good at this, and some individuals are absolutely not good at this at all, even to the point where they can't recognize their own face. Now, I don't know if Mr. Oliver Sacks would be able to see that this is a face, right? I don't know how much his prosopagnosia would affect his ability to see a face at all. Like, could he see that that's a face and he just wouldn't recognize it if it was somebody, or does he not even know what a face looks like? My guess is he would say, yeah, I can see that that's a face. I just wouldn't know who it was if it was supposed to represent a particular person. But again, this is why we can see faces in things that aren't really faces. This is why you might be walking around town and be like, hey, look at those weird dots on the wall. They look like a face, even though they're just weird dots, right? We see faces in everything, and we're meant to see faces in everything, and we think, evolutionarily speaking, that that brain region has become so important in human brains because it's important for bonding. That when we're still young and our brains are still developing, particularly our visual system is still developing when we're an infant, that's one of the very first things that we want to be able to see so that we know who to bond with, right? Our individuals' faces, because usually the caregivers are going to stare at you and look over you, and you want to be able to see, oh, there's their face. That's a safe caregiver. I should respond to them. All right. So that's some of the cool things that we can do and that our brain is doing to help us see. So how does that actually all work? Well, we know that vision is, for humans at least, our primary sensory experience. For most of us that have intact vision, we have way more of the human brain dedicated to vision than we do any other sensory system, but that's not true for every organism. We've talked about that several times before. For example, the rat brain has this huge olfactory system relative to ours. They have something, everybody has something called an olfactory bulb, which is partly where sense of smell is processed. But in the rat, their olfactory bulb is just huge, especially relative to the rest of the size of their brain, but also relatively compared to ours as well. And why? Because they use their sense of smell a whole lot more than their other organ sensory systems. And again, we don't, so ours is smaller comparatively speaking. So understanding the visual system's organization is also going to be key to understanding not only how our brain works, but again, a lot of human behavior. So let's take a look at how we see things. And the way we're going to start off with is just by talking about how our basic organ that does the sensation part works. So what do we actually see? Well, we need our eyeballs to do that. And what we are looking at, in this case, this eyeball is staring at a flower. So when it stares at the flower, light is all in the environment, and that light is going to hit this flower, bounce off the flower, and the light is then going to enter into the eyeball, and actually ultimately get projected onto the back of the eye, actually in an upside down two-dimensional way. And that upside down two-dimensional picture is ultimately going to be sent to the brain thanks to the cells that are in the back of the eye. The cells are going to convert that upside down two-dimensional picture into action potentials and are going to send that message in the form of a bunch of different action potentials to the brain. And then the brain is going to reconstruct all that information and put it all back together, re-add in the 3 dimensions, flip it back the correct direction, and that's how we were able to see what we see. So the actual perception part, the action potential part, is being sent as an upside down, two-dimensional, basically pixelated information. If you remember from the last lecture, I showed you that pixelated information. Yeah, that's what we're seeing. Right? That's literally the perception part that's going to the brain. So our brain is really what's doing all the fancy stuff that allows us to actually see what we see. Our brain is adding in the 3 dimensions. Our brain is flipping it the correct direction. Our brain is making it one complete image and not a bunch of separate individual images. Our brain is adding in the color, that sort of thing. All right? So our brain is doing a lot of the work that allows you to perceive and see what you do. The key here is the energy that we are detecting, and that is visible light. So light is just one form of energy. You heard that in the video. All right. So energy comes in a lot of different wave forms, all the way from gamma rays to radio waves, and we can only see a tiny little piece of it right here in the middle, stuck in between UV light and infrared light, which again, neither of which we can detect with our eyes. Our eyes can only detect what we have named visible light, which encompasses approximately 400 nanometers. That's the shape of the wave, up to about 700 nanometers. So if light at about 400 nanometers enters your eye, you're going to experience kind of a purplish or violet color. I think he said blue in the video. That's sort of true, but if you think about how we remember the rainbow colors, right? We said Roy G. Biv, red, orange, yellow, green, blue, purple. They always kind of leave the purpley colors off the end here, but yeah, it'll be a bluish purple color. And if light closer to 700 nanometers enters your eye, you're going to experience a reddish color, right? And everything in between is going to be a slightly different variation of color. Again, from that rainbow thing that you learned probably way back in like elementary school maybe, right? Roy G. Biv, you learned. And you can see all of those different kinds of colors. So that is the visible spectrum that you are able to see. Now, because light is traveling in wave form, all right, it's got some physics to it that allow us to see and experience some pretty cool things. But we'll get to that in just a minute. But let me just show you again how light is coming into your eye, and I want to talk to you a little bit about some of the properties of light. So again, in this case, this eyeball is looking at a key. All right, the light is going to go into the eyeball. All right, because of this lens behind the eye. All right, that information is going to get flipped upside down. That's how it gets flipped the wrong way and projected onto the back of the eye. All right, that information is where it's then going to meet up with those photoreceptors that we talked about. Right, that are the specialized sensory neurons that can convert it into action potential, and then that information is going to get sent to the brain. And so all of this sort of information here is talking about how, right, that process happens, and it's sort of comparing it to the same process of a camera. But most people have no idea how cameras like this even work anymore, unless you're a fine arts major or minor, you've ever taken a formal photography class where you actually used a camera like this that had in a built-in lens like that and actually had film and all that kind of stuff, because most of you just use your cell phone now and you have no idea how the world used to work back in the day. So I'm going to actually show you a slightly different version of talking about the parts of the eye and how this works. I might still talk a little bit about lenses, but I have actually pre-recorded this before, so you're going to maybe hear a slightly different tone to the video, but I'm going to talk to you about the parts of the eye, and I'm going to give you some basic definitions, and I'm going to talk to you about how this process works. So I'm going to skip ahead here into this video and show you sort of where I am headed. All right, so I'm going to talk to you a little bit about these terms here. I'm going to go through sort of the structure of the eye in this other video that I'm going to show you. I'm going to talk to you about the parts of the eye. I'm going to talk to you about the retina and then some special spots on the retina. And then I'm going to come back in just a minute and I will talk to you a little bit more about the structure of the retina and the specialized cells. So I will be back to this set of slides in just a minute. So just know that I'm going to add a tiny bit to your slides here. So I will be right back. All right, some other cool things. Because light travels in waves, there are two terms that we're going to talk about. So we're going to do a little bit of physics here, but because of this physics, you're going to understand why you can see the cool stuff that you can see. So we're going to talk about what wavelength means, and we're going to talk about what intensity means. Now, to give you the end of the story first, because light can travel in different wavelengths, you can see different colors. That's what I was talking about before, with the 400 nanometers being more purpley blue and the 700 nanometers being more reds and everything in between, greens, yellows, oranges, all those sorts of things. So because of wavelength, you can see color. And because of intensity, right, so how intense the light is, you can see brightness, the obvious one being dark versus light. But you can also actually see brightness that sort of mixes with color, and you can have very vibrant colors and you can have very pale colors. So intensity and wavelength can actually work together as well. So let's start with wavelength. Right? Wavelength gives us the ability to see hue. Hue is the fancy word for color. Right? Now, here's what wavelength is. These light waves, again, are traveling in waves. Just like in the ocean, you see wave after wave after wave. Wavelength is literally how long the waves are. So you would measure it from the top of one wave to the top of the next wave. And if it's a shorter distance, you're going to see something that comes more like bluish or purple. If the top to the top is a longer distance, you're going to see something more like orange or red. Now, you're also going to notice I have high-pitched sounds and low-pitched sounds here. You're going to see this information again when we talk auditory system, because sound works the same way. But for light, wavelengths, right, the top of one wave to the top of the next, shorter wavelengths give you bluer colors, longer wavelengths give you redder colors, and the wavelengths in between give you all the different colors that we can see. So one more time here, again, more towards the 400 nanometers, you're going to see purples and blues. Towards the 700 nanometers, you're going to see oranges and reds. Right? And again, you're going to be able to detect everything in between for the most part. Right? So again, wavelength gives us the experience of hue, which is the fancy word for color. All right, the second part, the second little bit of physics here was intensity. Intensity gives you the experience of brightness. Low intensity light is going to look very dull or dark. High intensity light is going to look very bright or brilliant depending on your kind of color. So again, intensity is measured with how tall the waves are. So again, wavelength was top to top. All right, intensity is how tall it is. If it's a lot taller, it's going to be a lot more intense. It's going to be a very bright color. All right, again, you're going to see the same thing for sound later. This would give you a louder sound. A smaller amplitude, right, or a smaller height is going to give you a duller, a softer color, or a more dark, like you'll experience that it doesn't look very light in a room. It looks very dark in the room. Does that make sense? This is called amplitude. Amplitude is how high the wave is. Now we hear amplitude, we more think sound, right? Like how many amps does this put out? That's amplitude essentially, all right? But it works the same way for light. So how tall the wave is will give us a different amount of intensity. The next thing that I want to talk about a little bit is the actual structure of the eye. I am going to stay on this picture for just a moment. I'm going to talk through some of the parts, and then I'm going to skip ahead, all right, and I'm going to show you some definitions, all right, and I'm going to let you write them down. But before I do that, I want to talk about the parts of the eye. Now, hint, for a quiz or for an exam, I might have you label the parts of the eye that I'm going to go over here. So you're going to want to practice knowing not only what the structures do,
right? But you want to be able to tell me where they are as well. All right, so this is an eyeball. This eyeball happens to be looking at a candle flame. And before I start talking about any of the parts, I want to show you something weird that your eyeball does. Look at this candle flame, right? Candles, you have the candle here, the flame is on top. If you turn it upside down, it makes a big mess because wax gets everywhere. So this eyeball is looking at a candle held upright, but look at what happens when that light comes into the eye and is projected onto the back of the eye. And I told you that's where the receptors are. Look at what the candle is. The candle is actually upside down, right? So here's something really cool about your visual system. The information that comes into your eye comes in upside down and broken apart into pieces. If you remember that bottom-up processing, right, where everything was broken up into pieces. Your brain, right, so that's sensation. Upside down, broken into pieces. That's the sensation that comes in. Perception, right, gives you the ability to flip it back the right direction and to see it in 3 dimensions. So your brain is actually doing a lot of the cool effects that you actually quote unquote see is done by your brain, not actually by your eyeball. I digress. There was a cool study that I did in college. I'll tell you about it very briefly. So when I was in college, they were doing this study. There's these glasses that actually more like goggles that you put on, that you wear, that flip the image of what you're seeing. So when you put them on, everything looks upside down. So basically I put these goggles on, and if I was looking at this candle, it would look upside down. All right, so if it came through eventually into my eye, it would be right side up on the back of my eye. Now that message is going to get sent to my brain, and my brain is used to doing what? Well, my brain is used to flipping what I see. So at first, everything that I looked at looked upside down. It was really freaky, right? And you weren't allowed to leave the lab. You had to sit in the lab. And what they were testing was how long it took for your brain to correct for the weird goggles that you had on. Let me tell you, this is the weirdest experience. So it took me several hours, I think it was almost 3 or 4 hours, before I, even with the goggles on, started seeing things upright again. And in between, as it was sort of figuring it out, things would like flip right in front of me because my brain was going, wait, what am I looking at? What is it supposed to be? Is it supposed to be upright? They wouldn't let you walk because if you can't even imagine how weird it felt. So to try to walk when everything looked upside down, it was just beyond bizarre, right? It was cool, but it was beyond bizarre. Right? So basically what they were testing was how long is it before the perception part of my brain figures out, wait, something's not right here, and flips it so that it was right. And then once my brain figured it out, it's no different as I just had on regular goggles. Everything looked normal. Right? But then it got weird again, because then they did the second half of the experiment, which was take off the goggles and see how long it takes my brain to revert back to helping me see right side up. Now, that part didn't take nearly as long. That part only took about 30 minutes. But holy cow, did I have the worst headache ever afterwards, because my brain was just like, what are you doing, woman? I'm trying to help you see correctly. Quit flipping things upside down in the wrong way. I'm trying to help you out. It was pretty cool. All right, let's talk about the parts of the eye. So this eyeball is looking at a candle, right? This candle is reflecting waves of light, and they're going to come into the eye. The first thing it's going to pass through is an outer coating, hopefully a clear outer coating. Sometimes it gets a little cloudy, not usually until you're older, but that clear outer coating is called your cornea. For any of you that are like me and wear contacts, that is where you're sticking that contact to. So when you open your eye and you put that contact on there, you're resting the contact on top of that clear cornea, right? The clear cornea is a little bit of a thick layer. It helps protect so that hopefully things don't get in your eye and cause damage. It hurts like the dickens if it gets scratched. Again, sometimes when you're older, it can start to turn a little bit of a darker color. There's some whole disorders there. But for the most part, for most people, your cornea will stay this nice, thick, clear image the rest of your life, right? It's going to help protect the eye. All right, right behind that cornea, right, you're going to see two of my favorite parts of the body. Well, you really only going to see one, but I'll explain what I mean in a minute. All right, you're going to see a colored ring in the eye. All right, some people have all different kinds of colors. There are so many different shades of brown and gold. There are blue, there are green, there are some that are kind of bluish green. Mine look kind of gray depending on the day. All right, there are tons of different colors. Right, that ring of coloredness in your eye, right, is called the iris. And it isn't just a ring of tissue. It's actually a muscle. It is the prettiest, in my opinion, the prettiest muscle in your body because it's usually some cool color. Again, some sort of shade of brown or black or gold or blue, right? So that's the cool part number one. The second thing that you will also see right behind the cornea is a big black dot. That black dot actually isn't anything. It's actually a hole. And that hole is called your pupil. So the iris is the pretty colored ring. It's a muscle. And because it's a muscle, it can expand and it can contract to make the hole or the pupil in the middle bigger or smaller. Right? And it does this on purpose so that you let in the correct amount of light. You don't want to let in too much light or you won't be able to see. If it's dark, you want to have it open really wide so that any and all available light can come in so that hopefully you can see, etc., etc. If you've ever gone to the eye doctor and they've given you some medication that purposefully dilates the iris, right? They'll say it's dilating the pupil, but again, the pupil is just a hole. So really what it's doing is it's dilating the iris so that the pupil hole gets bigger. Right? You can imagine how weird that feels when you go out into the bright sun. They usually will give you these really dark sunglasses to wear afterwards because your iris is sort of paralyzed temporarily, so it can't contract and close and make the hole get smaller. It also makes it really, really hard to see and hard to focus the light, which is kind of funny. So those are the two cool parts that are right there behind the cornea. You have an actual part called the iris, that's the colored part. You have a hole, which is called the pupil, and that iris stretches or contracts or gets bigger or smaller so that the pupil, the hole, gets bigger or smaller so you let in the proper amount of light. Why? Because that light is going to be passing through the structure right behind it called the lens. And just like the lens in a camera, the lens's job is to help focus the light and make it a clear picture. And where it is focusing it onto is the very back of the eye called the retina. So the entire back of the eye is called the retina. And it is the job of the lens to focus the light directly onto the retina because the retina is what's made up of all those specialized neurons. The retina is basically just a big layer of neurons in the back of your eye. Your lens is pulled by a series of muscles. You do not need to learn those, but because it's pulled by a series of muscles, it can be pulled and stretched, or it can be relaxed and bunched up, so that depending on how far away or close something is, all right, you can get it to exactly hit the back of your eye where the retina is. All right, so the whole back of the eye is called the retina. I got 3 more parts to tell you. All right, within the retina, so within the whole back of the eye, you have 3 specialized parts. One of them is called the fovea. You can see it a little bit here. I'm going to show you another picture later. It's actually a small indentation in the retina. It's sort of like a little ditch, a little hole in there. The fovea is the point of central focus. So if you want to look at something and you want to see it clearly, you're probably going to turn your head to look at it. Because what happens when you turn your head is your eyes line up correctly so that the light that the thing that's coming off the thing you want to see directly hits that fovea. The fovea has the clearest vision. It's the point of central focus so that you have the clearest vision. So, for example, if somebody goes, "Hey," and you turn and you go to look at them, right? You're going to turn your head, your entire head, and stare at their direction so that you can clearly see them. You're not just going to move your eyeballs a little bit. Again, you could probably see them a little bit, but you're not going to get as good and clear of detail because it's not going to be hitting the fovea. So the fovea gives you that clear vision. All right, another couple things that you're going to have. Remember, all of the cells that make up the retina and the fovea and everything here are neurons. All right. Now, there's specialized neurons that can detect light. The dendrites are specialized to detect light, but because they're neurons, they're still going to have a cell body, and they're still going to have an axon. Right? And that axon is going to be what sends the message eventually up into the brain saying, I saw something. Right? But again, you have hundreds and hundreds and hundreds of neurons here, right, in this retina. Each one of those neurons is going to have an axon. And instead of the axons leaving, right, in a whole bunch of places, they actually sort of all bundle up together and leave out of one location. We call that big bundle of axons that is leaving the optic nerve, right? If you ever have heard the expression, I pinched a nerve, right? If you legitimately did that, what that really means is you applied pressure or you kinked, right, a whole bunch of axons grouped together, because that's what a nerve is a whole bunch of axons that are running around in the same direction, going in the same direction somewhere. So all of your visual neurons, all of those axons leave the same place. They leave all in one big bunch called the optic nerve, and each eyeball has one. So each eyeball has an optic nerve that comes out to send information to the brain. But because of that, because all the axons leave in one location, you have what they call a blind spot. So you have this little tiny spot where there's just no room for dendrites. There's just no room here for cell bodies and dendrites. So that if light hits this part right here, you actually won't see whatever it is. Now, you have a couple things to your advantage. First of all, you have two eyeballs instead of one, and if both of your eyeballs are working, they're actually tag teaming. And what they're doing is they're constantly scanning the environment, whether you know it or not. They're twittering, they're twitching just a little bit constantly, right? They're sending the information to your brain, and your brain perceives, hey, there's a hole here, there's a piece of missing information, but it fills it in with the information from the other eyeball. So what you perceive is nothing. You don't normally experience your blind spot, right? There's a really cool trick I'll show you in a few slides. It's actually in your book as well. So you can test your blind spot, but you have to cover one eye to do it so that the brain can't fill in the missing pieces. All right, so those are the parts of the eye that I want you to know. I want you to know the outer cornea. I want you to know the iris, right, which has a hole in it called the pupil. I want you to know the lens. I want you to know the retina. And specifically on the retina, I want you to know the fovea, the optic nerve, and because of the optic nerve, the blind spot. So I'm going to flip to this slide. I encourage you to hit pause and write down these definitions. Let's talk a little bit more about the lens. So again, I've given you the actual definition here. If you need to pause and write down that definition, you can. But I also want to talk about what the lens is actually doing and give you a formalized word. So I told you when I was showing you the picture of the eye that the light comes through the cornea, goes through the pupil, which is the hole made by the absence of the iris, and it goes through the lens. And the job of the lens is to be pulled and stretched by a series of muscles so that the light exactly hits the back of the retina, right? That process of pulling and stretching and changing shape so that the light always hits the back of the retina is called accommodation. And if you want to test this, you can actually see and experience your lens in motion. Here's how you do it. You hold your finger up in front of you. So make like you're holding up the number 1 finger in front of you. All right, hold it about 6 inches or so away from about the middle of your face. All right, and stand facing a wall. Then what you're going to do is you're going to stare at your finger until it becomes crystal clear. And then without moving your finger and without moving your head, move just your eyeballs and look at the wall behind you. All right, behind your finger, until it becomes clear. Then move your eyes back to your finger until it becomes clear. Move your eyes back to the wall. Move your eyes back to your finger and back to the wall. And you can do that as many times as you want. But what you'll see is it takes it just a fraction of a second, right, or maybe longer if you have visual impairments like I do with my contacts. It also might take it a little bit longer if you've been having an adult beverage. It might take it a little bit longer if you're tired. There's several things that can take it a little bit more time for that lens to accommodate. But what you're going to see is that lens is going to be busy stretching and flattening and stretching and flattening as you're going back and forth, and your eye is going to be going, what are you doing? Stop doing that. But that's what accommodation is for, and you are constantly doing this as you are looking around the world. As you're looking at things close up, it's going to accommodate one way, and as you look at things further away, it's going to accommodate another way, and your lens is constantly stretching and pulling and moving in order for you to do that. And again, the reason that's important is because you need that light to hit the very back of your eye on the retina, because that's where all of those neurons are. That's where all of those specialized neurons with specialized dendrites are to detect the light. So let's talk a little bit more about the retina. Again, if you want to pause and write down the definition here of retina, I suggest that you do that. But the retina, again, is just the light-sensitive inner surface of the back of the eye. So the retina is the entire back of the eye. And when I was talking to you earlier, I actually oversimplified a little bit. There's actually a whole bunch of different kinds of neurons here. And if you find this stuff interesting, I highly suggest that you go on and take biological psychology, because you'll go into this in a whole lot more detail. But the general idea of what's going on here is that you have certain cells, and we call them rods and cones, and these are specialized neurons that are able to take the light and convert it into action potential. Those rods and cones pass it on to a couple other kinds of cells, and then the neurons of those cells, the axons of those neurons are what leave out that optic nerve. All right, so again, a little bit oversimplified what I told you before. There's actually tons of different kinds of cells. There's also some things called amacrine cells and horizontal cells. There's a whole bunch of kinds of things. All of them are different types of neurons. All of them are needed to help you see everything that you're able to see. But the number one thing that we're going to focus on for this class is the very first stop, which is the rods and the cones. Now, something else weird that I want to show you about the retina, right? As the light is coming in, so you can see here's the eyeball, here's the iris that's colored, there's the pupil, that's the hole. The light comes in and it hits the retina. Look at what the light has to do. The light actually has to pass through all of these other cell layers first before it hits the rods and the cones, the specific neurons that can detect light. That's a very weird phenomenon, right? But it's something that absolutely happens. But that's another thing that I'm going to make a point of in just a minute as to why your fovea is shaped the way that it's shaped. But the retina in general, the entire back of your eye, is made up of a whole bunch of different kinds of neurons, and the ones that we're going to learn for this class are rods and cones. All right, a little bit more about the optic nerve. So again, the rods and cones have passed their message on to various other kinds of neurons. And all of those axons from those neurons gather together and leave out one location called the optic nerve. So the optic nerve is what's carrying the neural impulse, or that action potential, from the eyeball itself to the brain. Because of the optic nerve, you also have a blind spot. So there's that definition there. And again, if you need to pause and write these down, that's fine. But as I also told you, you have one spot on your retina called the fovea, and that spot gives you the clearest vision. So again, if you wanted to see something very clearly or pay attention to something, you probably turn your entire head to look at the object, because that's what's going to help you to see it the most clearly, because that's what allows the light to actually hit that fovea. And I also told you the fovea was sort of indented or made like a little ditch or a little hole. Here's why, right? There's two reasons why the fovea gives you very, very clear vision, right? The first one is because of this indentation. All of those other kinds of neurons that I told you about that were there, so if you go back, remember all the light had to go through all these other kinds of neurons? In the fovea, those cell bodies of those neurons purposefully grow sort of to the side. They almost leave an extra area of unexposed, right? Rods and cones, right? So this is what makes it easier. So the light doesn't have to go through all of these cell bodies as much. It can just go straight there and interact with those cells that are there. And that's the first reason why you get such clear vision in the fovea, is because of the way that the cells grow. I'll tell you the second reason in just a minute. As promised, here is a test for your blind spot. Now again, this might be a little hard to do on the computer, and if you are going to do it, you're going to need to pause the screen so you can do it. But what you need to do is cover your eye, all right, and then move your head closer or further away from the computer screen while staring at the black dot. And you'll get to a certain point where you will not see the car anymore. Now, if you keep both eyes open, it's not going to work because your brain is going to use its cool power, all right, of perception to fill in the gaps. But if you close one eye and don't give the brain that ability, you will actually find that the car will disappear. That's your blind spot. And again, the reason you don't see it there is because when you get in that exact spot where you can't see it, the light that's bouncing off your computer screen from the car is landing on the optic nerve in your eye, which is why you have a blind spot. So test that out for yourself. All right, a couple other things that I want to talk about. I told you the two kinds of cells, the two kinds of neurons I wanted you to remember were rods and cones. Let's talk about these two types and why you need two types of cells. Now, collectively, they are called photoreceptors. Photo is a fancy word for light, so it literally means these are light receptors. So those specialized dendrites are what are receiving the light energy. And I really wasn't kidding when I said that you have tons and tons and tons of neurons in the back of your eye alone. Cones, you have about 6 million. Rods, you have about 120 million. And that's just the rods and cones. That's not all those other neurons that I was talking about. You have tons of neurons in your eyes. But here's the second reason, right, why the fovea, right, is so clear. So not only is it because of the way the cells grow, but it's also because that's where you find the majority of the cones. The cones are located in the center of the retina. That's the fovea. They're not very sensitive in dim light, but cones are responsible for helping you to see color, and cones are responsible for helping you to see detail, right? So if you wanted to see something in fine detail, if you want to see the color of it, you need to turn your head, you need to directly stare at it so that the light hits that fovea, that center of the retina, so that it can interact with your cones. Now, what are the rest of the cells doing? Well, the rest of the cells, and actually it's most of them, 120 million of them, are located in the periphery. So they're in kind of the rest of the retina, right? They are highly sensitive in dim light, which means that even when it's dark outside, even if you can't quite tell what color something is, right, you can still see the overall shape. You can see if it's moving, right? So rods don't give us color and they don't really give us detail, but here's why they're important, right? If I'm standing outside and all of a sudden an object comes towards my face, what do I need to do? Well, I need to do several things. I either need to duck or move my head, put my hands up if I can. If nothing else, at least close my eyes so I don't get damaged. All right? All right, but does it matter if it's a rock or a baseball that's coming at my head? Do I need to know which one it is before I decide, hey, I should block my face, or I should duck, or I should move my head? No, doesn't matter at all. The point of our rods is that they are quick responding to motion. All right, not just because stuff is coming at your face, although that's a very good example, but our rods help us to tell if something is moving and the overall shape of it. So I can say, oh, this is some kind of object and it's coming towards my face. I need to do something about it. And it's only later, right, that you're going to look on the ground to use your cones to go, oh, that was a baseball, or oh, that was a rock. All right, because again, in the heat of the moment, it didn't matter what it was. You just needed to get your face out of the way so you didn't get damaged, you didn't get hurt. That's what the rods are for. Motion, detection, how something's moving, how fast it's moving. All right, now combine that, because you have both, we're typically able to see very complex things in a very complex way. So those are the photoreceptors. Those are what I want you to remember. Okay, so we've got a whole bunch of neurons. They're sending their axons out of the eye via an optic nerve. So here are two eyeballs. Each eyeball has an optic nerve, right? Those optic nerves, right, do a little bit of crossing over, right? And it will eventually go to the thalamus, and then will eventually go to the back part of the brain.
Okay, so I am back with the slides that you have available. Again, I was showing you a previously recorded video partially so that I could give you some additional slides and show you some additional images that I had, but I wanted to return to the slides that you have available to make a few additional points. So again, in that previous video that I had shown you, I had said that I wanted you to know the rods and the cones, and that is true. I do want you to know what the rods and cones do. But also for this class, I want you to know one other kind of cell, and that is the retinal ganglion cells. And I think in the study guide, it just calls them ganglion cells. Again, either way, they're the same thing, retinal ganglion cells or ganglion cells. And the reason I want you to know these retinal ganglion cells is because these are the cells that send the axons out that will ultimately be the optic nerve that create that blind spot that I was talking about. So I was just generically in that previous video saying all of those axons come together to make the optic nerve. Well, what I was really meaning was all those axons of the retinal ganglion cells. So the rods and the cones are the first cells that interact with the light and originally produce the action potential. And then you have neural relay. So the rods and the cones pass their message on to bipolar cells pass their message on to retinal ganglion cells. Then you've got some other sort of helper cells in between. You've got some horizontal cells and some amacrine cells. And again, if all of this information is interesting to you, I do highly suggest that you go on to take biological psych. At UNC Charlotte, it's called physiological psych. That's the 3113. But again, the last stop in the actual eyeball is called the retinal ganglion cells. And they're the ones that have these really long axons, right, that ultimately will all leave as one big bundle. Right? And that's another trick for you. An axon that joins up with a bunch of other axons and makes a big bundle, that's where we get the word nerve from. So if you've ever pinched a nerve, what that means is that you pinched a whole bundle of axons. But anyway, that's where you get your optic nerve from. All right, so the light comes in, stimulates the rods and the cones. They produce an action potential because of that light interaction. They pass their message on, again, using neurotransmitters. The bipolar cells then produce an action potential. They pass their message on with neurotransmitters. The retinal ganglion cells produce an action potential, and that action potential travels down the big bundle of neurons, the big bundle of axons, called the optic nerve, up into the brain. Right. Again, you saw this in the video, but here's a slightly different way of looking at it. The fovea gets the clearest vision. The first reason is because all of those other cells, so those bipolar cells, those retinal ganglion cells, and all those other things sort of grow to the side, so you get this indentation, and all of the cones are exposed. We talked about that and how that gives you the clearest vision. So how I remember what cones do is that cones give you clear vision, and that starts with a C, and they also are in the center of the fovea, of the retina, and that starts with a C. So color, clear, center, all of that is where cones are located. So that's how I remember what the cones do. So that's one of the reasons why the fovea is such the clear vision there. So again, that was the start of the discussion of the receptors. I already talked to you about the receptors in the last video, but again, here is more information. So just add that together from what I talked about before as you're taking your notes. And again, here's the second reason why you get such clear vision in the fovea is because your cones are there. So again, the fovea has clear vision because of the cones. So cones, clear, center, all of those C words go together. All right. Cones are shaped because they got kind of a cone ending. Rods are shaped because they have kind of a rod ending. We're not super creative. Again, cones give you color vision because they absorb various light at different wavelengths. right? And then depending on which wavelength of light they absorb, it will ultimately be sent to the brain, and the brain will help you experience it as a slightly different color. And then depending on sort of the combination that comes in, it will allow you to see different colors. There are approximately equal numbers of red and green cones, and there are fewer blue cones. So you have three main kinds of cones, at least according to the trichromatic theory, which is one of the two major theories of color vision. And that theory says that you have three main kinds of cones, ones that detect blue wavelengths of light, which is short wavelengths, ones that detect green, which is sort of the middle wavelength of light, and ones that detect red, which is the longer wavelengths of light. Again, you have about equal numbers of red and green cones, slightly less numbers of blue cones, but again, depending on how many of each cone gets activated, again, your brain will interpret that as experiencing a certain color. Again, we talked about a bunch of different types of neurons in the retina. Again, I want you to know, right, three. I want you to know the rods and cones, and I want you to know the retinal ganglion cells. But again, just for your knowledge, I gave you some definitions of the other three kinds of cells that are there, but I'm not going to necessarily ask you about those other three, but I did give you a definition for the retinal ganglion cells here. And again, the big job that they are doing is taking the incoming information from the other cells, right, and sending that information out via their axons, and it's their axons that produce the optic nerve. All right, so again, they're sending out their axons, right? Ultimately, it's going to go to the thalamus. So we're moving into that neural relay part here where we're sending the information to the thalamus, right? You saw in the video too how there is a crossover, right? That crossover is due to the visual field. So the visual field, right, from this part of the visual field is going to go to the opposite side of the brain. This part of the visual field is going to go to the opposite side of the brain. You're going to see this pattern a lot, right? That one half of the body, or one half of the visual field, one half of the auditory field is going to get processed by the other half of the brain. Same thing with sort of like touch and movement as well. It's going to get controlled by the opposite side of the brain. So for example, movement on one side of the body is controlled by the opposite side of the brain. And sort of the same thing with vision as well. It's just a little bit weird when you think about your eyeballs. It's not one eyeball that's being controlled by the opposite side of the brain. It's one visual field. So this visual field gets controlled by the opposite side of the brain. And that visual field comes from this side of the eyeball. All right, so this visual field goes to that side of the brain, and this visual field goes to that side of the brain. All right, I also briefly talked about something called the optic chiasm in the last video. I'm going to go into a tiny bit more detail here because that's where we are leading up into heading to the thalamus. So again, what you have going on is once the neurons leave the actual eyeball, so you have the axons from the retinal ganglion cells leaving through the optic nerve, they are actually going to do that crossing over so that the visual fields can correctly be processed by the correct side of the brain. All right, that crossing over happens at the optic chiasm. So that's where you get them going to the proper side so that the left visual field can get processed on the right side of the brain and the right visual field can get processed on the left side of the brain. Once that crossing over has happened, you're going to get those two bundles of information going to the thalamus. And the specific part of the thalamus that processes visual information is called the lateral geniculate nucleus of the thalamus, which sounds like a weird word, right? But you're going to hear something similar when we get to sound information about the thalamus. But again, for vision, it's the lateral geniculate nucleus. All right, so we've reached the thalamus. So now the thalamus is going to send that information up to the cortex. This one should be easy to remember because, again, there's an entire lobe of the cortex for the primary sensory cortex, which would be, excellent, you remembered, it's the occipital lobe. So Sometimes it's called the occipital cortex. It's the exact same thing. You have an entire lobe, the occipital cortex, that is dedicated to vision. So the entire back occipital lobe is dedicated to vision. Now, you have one little slice of it that is technically called the primary visual cortex. Sometimes it's also called the striate cortex because it kind of looks striped. But that's where you're going to originally, that information that's coming up from the thalamus, particularly the lateral geniculate nucleus of the thalamus, is going to come to. Then you've got immediately next to it on either side what we would call the secondary visual cortex. But you've actually got several regions of secondary cortex. We call those regions V2 or visual 2 through visual 5 kinds of cortex. And they are responsible for processing all different kinds of features of your vision. So all the different cool parts of the vision that you're going to need to sort of pick apart, because remember, what went into your eye was an upside down, two-dimensional, pixelated version of what's actually happening in the world. And so your eye is going to have to reconstruct everything, put it all back together, add 3 dimensions, add color, add movement, whatever it is that it needs to do. And so you're basically going to have to reconstruct all of that with all of this kind of secondary parts of the visual system. Right? So you've got your primary visual cortex, right? And then you've got sort of 2 pathways that start to emerge, right, from the striate cortex. But again, here's sort of why it's called striate cortex. See how it's kind of striped? That's literally why we named it that. Again, we're not super creative as scientists. We don't come up with super creative names. But here's the occipital lobe. Again, sometimes it's called the striate cortex. All right. What you're going to start to see is that as vision starts to be processed by some of those secondary cortexes, and then eventually it's going to be processed by association cortex, is that you're going to have some of that information that's going to ultimately be sent this way towards the parietal lobe, and some of that information is going to be sent this way towards the temporal lobe. And the reason it's going to be sent in those 2 different directions is because it's going to be processed in 2 different ways for 2 different kinds of information. All right, so let's talk about that. The dorsal versus the ventral stream. The dorsal pathway is the pathway that went towards that parietal cortex, right? And that pathway is what we call the how pathway. That pathway is going to help you to process kind of the movement and the action part of the whatever it is that you're seeing, right? So it's basically guiding information from originally the occipital cortex towards the parietal cortex, and it's giving you information about how something is moving, right? Or how you might need to move if you want to interact with something. So if, like, for example, I have a cup sitting there on the table and I want to move my hand to go pick it up so that I can drink it, it would also allow me, as I'm visually watching my hand and the cup come together smoothly so that I don't knock the cup over, so that I don't miss and not actually grab the cup, so that I can smoothly grab the cup and then move the cup back towards my body. All right, so the dorsal visual stream is giving information about how something is in my visual system, how it's moving, where it is, exactly what's happening. The ventral stream, the one that's moving towards my temporal cortex, is giving me a lot of information about what. So if you remember way back in lecture 1 when the crash course video was talking about the guy that couldn't recognize faces, this is where, again, part of the pathway was it helps you identify faces is. This is where you would find that specific brain region. And it's a specific kind of what, and the what would be faces, right? So you've got a lot of different parts that run along that ventral stream that are allowing you to identify very specific kinds of things. Again, an example would be faces that allow you to identify specific kinds of objects or specific features of objects. So again, this is what we mean by secondary processing and association processing, is you're putting stuff together with other kinds of pieces of information, including memory and other kinds of sensory information, to make sense of what you're experiencing in the world. So the dorsal stream is processing one kind of information, that's that going towards the parietal cortex, and then one kind of information being processed, again, we call the ventral stream, as it's heading towards the temporal cortex, that's giving you other information. This is just it in picture form. Again, the information coming from the occipital lobe is processed again in two different ways because you need two different kinds of information about it. You need how, or sort of where, and then you need the what kind of information in the ventral stream. All right, the last bit of information in this lecture you heard a tiny bit about already and in the former lecture that I gave you, but I wanted to talk to you a tiny bit more about our ability to see colors. So the other reason I showed you that lecture that I did is because we didn't do a lot, I'm sorry, my screen is messing up here. We didn't do a lot in this lecture about giving you the physics behind why we are able to see colors, and you don't necessarily need to remember it for the exam, but I wanted to talk to you a little bit about the physics of why we can see colors. So partly the reason why we can see colors is because colors travel in wave form. And so you had this idea of wavelength and amplitude of the waves, how long the waves are from peak to peak and how tall the waves are, which gives you sort of this brightness, right? So you get brighter kind of pale, color versus deep dark color. So you get all that as a range of colors. So for example, even with just like blue, you can have dark blue all the way to light blue, or you can have all the range of what we would call like the rainbow colors. So if you take all light together, it sort of looks whitish, and you can split it with a prism and you can get all the different range of wavelengths of colors from red all the way down to kind of purplish. Again, the absence of any incoming color is going to be experienced as blackness. So again, there's a lot of different ways that your brain can kind of be seen. Now, what you're looking at here, right, is actually what would happen if you don't actually have all of your different photoreceptors, so those cones, working correctly. So I told you already in the lecture that there were three major kinds of cones that are responsible for detecting different wavelengths of colors, and then depending on sort of the ratio of how they get activated, your brain perceives that as a certain color. If you have, again, what we call a trichromate, which means you have three different color cones that you're able to see, you would see the normal range kind of the rainbow prism of colors. But if you're missing, or you have one of the kinds of cones that doesn't actually get activated correctly, you would not see all of the range of colors very well. So if you're lacking red cones, or if your red cones don't work very well, you may not see this image in the same way. It might look more like this. If your green cones don't work as well, it might look more like this. If your blue cones don't work as well, it might look more like this. Again, there are certain variations in here. People can be colorblind, which is actually a sex-linked trait, which means if you are going to have it, you're more likely going to be male because the copy that you need that would determine whether or not you're going to have the gene messed up is on the X chromosome, but it's on the part of the X that if you are a male with one copy of an X chromosome and one copy of a Y chromosome that made you genetically male, it's on the part of the Y chromosome that's missing that makes it a Y, right? So you would not have a second copy. So if you have a one disrupted copy on your X, you don't have anything there on the Y to recover from it. Whereas if you're female and you have one copy that's disrupted, as long as you have a second copy on your other X chromosome that is okay and intact, you're not going to be disrupted. But you would thus be called a carrier because potentially you could pass on the disrupted or the correct copy. It's not that women can't have color blindness. That just means that they have to have both X's disrupted. So anyway, that is what color blindness would mean. The most typical color blindness issue is going to be either a disrupted red and green cone combination. Blue is not typically the one that is messed up, although it absolutely can be. But again, humans are trichromates, meaning we have three sets of cones. Not all animals are trichromates. For example, rodents, rats, are what we call dichromates, which means they don't have all three and they don't necessarily see the color red. And I took advantage of this in graduate school. I ran some experiments using a red light in a room because I can see in red light because I'm a trichromate, but the rats could not. And for them, it may as well have just been dark. It may have looked sort of grayish to them, but it definitely didn't look red or very bright to them. And so for them, they didn't feel as disrupted because rats are nocturnal. They like to be awake at night. They don't like to be out during the day. They feel very scared and skittish when it's daytime. So it worked very conveniently that I was still able to see and do what I needed to do, but that they weren't disrupted by bright lights, that we could work in a red environment. So I kind of took advantage of the fact that they were dichromates and they were only able to see two colors. But anyway, let's talk a little bit more about seeing colors here. So again, color blindness is a genetic disorder, meaning you do have to actually have a disrupted gene. It is typically that you're going to have disrupted reds and greens. This supports that trichromatic theory that you have three different kinds of cones. This is the test that you might be given if someone suspects that you have red-green color blindness. They're going to give you something like this, and they're going to say, can you see a number in here? And basically what this is a series of a bunch of dots, and some of them are reds and some of them are greens. And again, if your color vision is intact, you'll be able to see the number 74 or 45 here. And if your cones are not intact, you would not be able to see those very well. Now, again, if you can't see them on the computer screen here, don't worry about it. You get a bunch of disruption with the way the computer screen displays colors and things, that this is not at all diagnostic. Again, if you have a suspicion that you might be colorblind, you've probably noticed it way before now, but you would also want an actual doctor to diagnose you in a formal situation, not with something like this over a computer screen. But just so that you sort of get the idea, this is the kind of test that they would use in a formal situation. So quickly, let's review that trichromatic theory one more time. This is one of the explanations of color vision. And again, it's the idea that we have three different primary colors that we see because we have three different kinds of cones that are sensitive to different wavelengths of light. So we have short cones that are sensitive to short wavelengths or blue light, cones that are sensitive to medium or green light, and then cones that are sensitive to long wavelengths or red light. And again, that's why we are trichromats. And then again, what we see, again, all of these cones are simply just sensing the different wavelengths, in other words, producing action potentials when they're interacting with that wavelength. But then what our brain does when that information comes in is, depending on the ratio of incoming action potentials, your brain will perceive it as different colors. So if you have predominantly incoming action potentials from blue cones, you're going to perceive it as blue. Predominantly medium cones, green. Predominantly long cones, red. And then different ratios of these, you'll perceive it as different other colors. So purple, yellow, orange, etc., etc. So this can help explain things like color blindness, but what it can't explain, and why we actually think there's two different theories that are working, and why prior to now there's actually been a whole bunch of fighting going on between the two different theories. I'm right, I'm right, I'm right. And finally we've decided, okay, we think you're both right, chill out. Is that actually there seems to be four basic colors. Yes, we think there's red and green, and yes, we think there's blue, but we also seem to think that there's yellow. And the reason we think there's a fourth basic color is because of this idea of after images. So if you were ever to go into a room, so for example, when I was working with those rats in the red room, right, where I had the red light on, when I would come out and I would go into a world with normal light, which was a mostly white light, everything would sort of look greenish for a while. I would sort of see kind of the opposite color for a while. It was kind of weird. And then my eyes would sort of readjust. And the opposite would be true, right? If you were ever to go into a room, for example, with a blue light, you would come out and things would sort of look yellow for a while. Or you go into a room with a yellow light and things would kind of look blue for a while. So we know that after images are a thing, and that can't be explained here, right? That just didn't make sense. And so there was this second theory that help to try to explain this idea of after images. And that second theory is called the opponent process theory. And it's been around a while, just as long as about the trichromatic theory has been around. And again, they fought for years and years and years to try to prove themselves right, to disprove the other. And again, where we are now is that we think both are right. We think both of these are true. But the opponent process theory thinks that basically you also have cones that are sensitive to red or green and blue or yellow, right? And that because of that, that's why you get this sort of after effect, right? That they respond to particular wavelengths, they don't respond to other wavelengths, and that's why you get sort of that after effect, that if they've been exposed to red for a while, when you take the red away, they kind of get hypersensitive to the other, or they sort of have this hypersensitivity to the opposite color. So opponent process is almost like opposite process, is almost kind of what they're saying here, is that you have these cones that almost have opposite effects with each other. And actually black and white kind of works the same way, although you don't really need cones necessarily for that. So basically what they're arguing is that you have cones that are sort of sensitive to these opposite colors, and that again, if one has been highly activated for a while, if you quickly take it away, you might suddenly experience a little bit of overactivation of the opposite color for a while. And I'm going to show you an example of an after image effect in just a minute. But this is actually important for another theory that you'll talk a lot more about again if you go on to take 3113, called color constancy, which is why you can still perceive color as long as you are kind of able to relate it to other surrounding colors, regardless of changes in illumination. Remember, basically, no matter how light it is in the room or how dark it is in the room. So some of those kind of optical illusions that were going around social media, like what color is this dress, or what color is this shoe, or whatever it is that were going around, that was sort of playing on this idea of opponent process theory, but it was picking at the idea of color constancy. All right, that if something looks red compared to something else next to it, like if it's next to something that's yellow or blue or whatever, right? Even if you turn the lights down, you're still going to be able to tell it's red because it's up next to those other colors, right? Your brain is sort of good at kind of comparing to what's around it. That if the lights are turned down, I can still ask you to pick out the red marker or the green marker or the yellow marker if you're able to compare it to other things that are around. Or if you go into a room and there's a red light on, you're probably still going to be able to pick out the green marker as long as you can compare it to the other colors that are around it. But if you're not able to compare it, you're going to have a much harder time because you're not able to sort of do this sort of comparison thing. All right, so here is this idea of demonstrating the opposing color pairs. So what I am going to do here is have you stare at this image, and then I am going to quickly flash up a white background, and what you should be able to see is an after image effect. So to make this work the best, you need to stare straight at this dot in the center, sort of relax your eyes, all right? And what you're going to do, right, is you're going to see the opposites a little bit. So the green should become red, the black should become white, the yellow should become blue, all right? And hopefully you'll be able to see the after image. So stare at it just a minute, all right? It's actually going to take about 30 seconds or so of staring, and then I'm going to bring it up. So bear with me. It's going to be a weird, awkward silence, and then I'm going to bring it up. Did it work? All right, if it didn't work, no problem. Again, the computer is also kind of weird. But if it didn't work, you can go back and try it again, and you can just pause it on this screen until you're ready, right? And then you can flip it again. All right, so that is the end of our lecture. Hopefully that makes sense. Hopefully you learned something about vision, and I will talk to you soon.